It's a real pleasure to have Alex visiting us. Well, she's spending the year at U Chicago this year, but she's from Paris. She lives in Paris. She's at the École Normale and leads a group broadly in biophysics at in the physics department there. She did her PhD in San Diego on protein folding, spin glass.
Gene regulation.
Gene regulation, and then was a PCTS fellow at Princeton, where she worked with Bill Bialik and Kurt Callan, and then started at École Normale and has been there for a while now. She's, has all sorts of awards and things like that, but she's an APS fellow as well. She's a senior editor for eLife. She's just one of the leaders in this field, and her work, the sort of different aspect, I think she's one of the few people that sees, I think, by the realm of biophysics very broadly, not just limited to sort of material biophysics or sort of literal physics, but sort of interpreting it sort of more generally. She's done, I love her paper on flocking, where she uses maximum entropy models to sort of study Starling dynamics. And she's done a bunch of work on gene expression, information flow and gene expression networks. But I would like to single out her work on what she will present today. I don't know if she's going to do the whole background, but at least speak on the topic, which is on immunology and quantitative immunology, and in particular, the generative process of the immune repertoire. And it's really impacted the field. Not just, by that, I don't mean physics of living systems kind of field. It's really impacted immunology. It's given that community a way to think about the generative process of the immune repertoire. And I think it just shows you sort of the standards can be sort of even higher. We can all sort of try to aim to do that. Of course, I failed to do that, but she really has and continues to. And so it's a great pleasure to have you, Alex. She's, by the way, around for the whole year. So if you sort of find that, oh, maybe you want to hang out and talk more to her, you can email her, and she's just sort of on the other side of the city. Welcome.
Thank you. Thank you, Hannah, for the invitation. Is there a stick or something?
No, no stick.
No stick.
No stick. It's great time.
Would you like that?
Yeah, that would be great. Very, I didn't think of that before. So, yeah, maybe Madhav should have told me that I could have talked about birds and flocks and stuff, but I'm not going to do that. I'm just not going to do the whole sort of immune background thing, since I did it on Zoom about a year ago. So this is going to be a talk about the immune system. I'm assuming you're not specialists in the immune system. Okay, good. Nobody? But, then I do this, and then people who actually work on the immune system get annoyed that I simplified things and didn't go into detail and didn't do justice. So I'm yeah. All right. So this is a collaboration of work we've been doing with Thierry Morin, who'll be here in a few weeks, and maybe he'll talk about birds. And flocks, and with a lot of other people that you see here on the theoretical side and the experimental side. So let me just go into it and sort of tell you what I'll be talking about, which are adaptive immune systems. So after, all our pandemic stories, you're probably aware that you have an immune system, an adaptive immune system. It's made out of B and T cells. These are cells whose job is to recognize foreign pathogens. So they basically have these receptors on the surface that I need a okay. I don't need it for changing things. I just need it for pointing back. So I think that's okay. Good. All right. So they have receptors on the surface, and these receptors physically bind bits of molecules and figure out whether whatever the binding is natural to your body, which we'll be calling the self protein because it's your own self protein, or it's foreign to your body, in which case it's most likely a pathogen. And so we'll be talking hopefully both about T and B cells. I hope I'll get to that. Right. Fine. So what happens when one of these cells recognizes a pathogen? Then it proliferates. It makes many copies of a cell that is identical to itself. And so that increases count. But when it doesn't, it dies. So you see there's some sort of dynamics. There's some nonlinear size control of this repertoire. Basically, this ensemble of different cells is called a repertoire. each cell has one type of receptor. So to recognize different pathogens, you need many different cells. And so you have this ensemble of different cells whose counts or frequencies go up and down depending on your past infections. So basically, it's like you're walking around with this personalized medical record. So, you know, when you go to the doctor and they ask you, did you have chicken pox when you were 7 and you don't remember? Well, most of us do remember because we have scars. But anyway, let's say you don't remember or you want to lie. Well, in principle, if we knew how to read this, you couldn't lie. Right? They just take some blood and they figure it out. So that brings me to the next step. This is the sort of experiments I'll mainly be talking about, is that you go in, you get a blood test, they sort out these T cells or B cells, and then they sequence these receptors. And since the receptor, as I said, one type of receptor per cell, that's sort of equivalent to knowing what kind of cells you had. So if we knew how to now make the mapping between receptor and what it recognizes, we're in business, okay? But we're not there yet. But that's sort of the deal with this field. So I'd like to talk about some more theoretical concepts today about how this whole thing works. And the first thing I want to talk to you about is this problem of self versus non-self. So remember, these cells, and now we're going to focus on T cells. Everything I said so far is true for both T and B cells, but now I'll say something that maybe will only be true for T cells. But the general problem is similar for B cells. So basically, the name of the game is you have a T cell who recognizes pathogens, some set of pathogens. And so we're going to think about it in an abstract phenotypic space that is called shape space. It was the fourth concept was invented by Alan Perelson quite some time ago. And so basically think about it the way it's drawn here. A receptor recognizes some set of pathogens which are within some bubble from it and doesn't recognize anything beyond it. Then you have many receptors and the name of the game is to cover space, right? So that you recognize all the pathogens that are out there. Easy so far? Okay. Now comes the but. You don't want to recognize your own self proteins. So you basically draw circles where you don't want anything recognized. And so you want to cover space, but avoid your own proteins. Simple, right? Already not, because if anybody's worked on covering, you know, you're already getting into difficulties. I, of course, haven't told you anything about the dimensionality of this phenotypic space that we know nothing about. But hopefully this is a problem that, you know, you've encountered in some form in your own research, because, you know, it's a pretty general setup. All right. So where we go, so people have been thinking about this for a long time. And this is, you know, this is how the pictures, basically the problem is presented. Well, maybe not within this discrimination problem, but, you know, that's what we know. What we wanted to get at is, given that we now have these sequencing experiments, we wanted to ask whether we can find something in the sequences of these T cell receptors, some features, for lack of a better word, that discriminate between foreign and self peptides. Basically, that, you know, if I look at all the receptors here that actually exist, can I figure out, you know, what is it in their sequence that makes them recognize this and not recognize that? Can I find some signature? Okay, so now sort of the textbook picture, what happens? T cells first get generated in a process which is called VDJ recombination. It's a random process. So the first thing to realize is you have of the order of 10 to the 9 different receptors. If these are proteins are encoded in genes. If you wanted to encode 10 to the 9 proteins in genes, this is way more genes than you have. You have of the order of 10 to the 4, 10 to the 5 genes, okay? So that's basically not how it works. There's a very interesting process that I won't tell you about, because I told some subset of you a year about, and we've worked on, so just trust me, we know how this is generated. We know a process that can generate actually way more than 10 to the 9th different receptors without hard coding them. So, but when they're generated, if you generate stuff like that, you also generate garbage. So basically you need to get rid of the garbage, and this is done in a process which is called thymic selection. Thymus is an organ that I believe is somewhere here, and there's two things that happen to it. There's first what's called positive selection. So the receptors get expressed on surfaces, and in the thymus, you only have your own proteins. So this is, think about it like school, think about it like an exam. Like you've trained, you've generated your receptors, and now they're being tested, but in a comfortable environment, meaning that if they screw up, nothing bad will really happen, right? They'll fail, because they're only being presented with their own with your own body's self-peptides. And the name of the game is that so first of all, if they don't bind to anything at all, that probably means they're misfolding, they're not a good receptor, they're not being expressed probably. There's probably some functional problem with them, right? If they really don't start sticking at all to anything at all. So that's this step, you get rid of those. And then there's the harder part, the extra credit exam, which is called negative selection, where again, they're being presented with only your own proteins, but if they bind too strongly to them, that's bad too, because that means you're going to have receptors that will bind really strongly to your own proteins also later on in what we call the periphery, basically in your bloodstream. So they'll be attacking you constantly and triggering immune response. Get rid of those two, okay? So basically so that seems, again, sorry, that seems simple. You get rid of the, this, we won't be talking about this, but you get rid of the ones that bind too strongly from self. So now we only have proteins that won't bind at all to your self proteins, right? Okay. Of course, this is biology and nothing is so simple. First of all, we know that this process is leaky. We know if we look in the bloodstream, we know there are self-reactive T cells, and some of them even have a function. They're called regulatory T cells, and they're sort of like a safety guard. So when a T cell gets overexcited and starts shouting, hey, infection, let's kill the pathogen, there's a subset of these cells that actually say, hey, calm down. And what they do is they're actually known for recognizing your own self proteins. And they do trigger this sometimes when a T cell sort of accidentally mistakes your own self proteins. But basically, what's important for here and now is that there are some types of T cells, and not just Tregs, but normal cells also, that we know are in the periphery and have managed to get through this test. The other thing, which is sort of more quantitative, and this is an argument that comes from my Chakraborty colleagues, is that this whole thing takes 4 to 5 days. So if you do a sort of back of the envelope estimate, and this is really a back of the envelope estimate, you end up with that in this time, each T cell is only able to probe 1 to 5% of self-peptides, just a few percent of your self-peptides, okay? So there's different ways of doing it, how many cells, APCs are antigen presenting cells, so how many cells a given T cell actually encounters that are able to present these proteins, how many peptides are presented, and so on. It's a cell consistency argument, but at the end of the day, basically it's an argument of limited time. If this were to work, every T cell would have to see every possible self-peptide. And if you think that it takes a few seconds for this interaction to happen, you have a few days, you basically, you think how many genes you have, so how many proteins you have, you don't have the time. Okay, maybe this isn't an exact number. This is the number that the Roop and Maran Kadar came up with, but it's of the basically not enough time remains true. So how systematic is it? And again, can we see signals in the sequence? So what we did,
excuse me?
Yeah.
So is it looking for any self-peptide or only like the surface proteins?
No, any. So basically, so what happens normally is that basically most of your cells constantly present what they're making to the outside, and the immune cells constantly probe it. In the thymus, you might be worried, well, why would everything be expressed in the thymus, right? We know that different cell types express different types of proteins. So there's a special enzyme that upregulates production of things that shouldn't normally be expressed in the thymus just for the purpose of this presentation. Okay, so it's really, and there's an active presentation mechanism in many cells. Okay, so the idea is that really for this to work, every T cell should see every self-peptide out of you. Okay, so what we did with Neil Friedman and the Weizmann is we did what immunologists do, is we built the mouse. And the basic idea of the mouse is that from this mouse, we will get both positive and negative examples. We will get cells that both passed and failed a specific test, okay? In the broad sense of passed and failed this thymic selection step. We'll break it down into a few more details. So this is actually what's called the spleen, is another organ, but it's basically a shortcut for what you see outside of the, after the whole process, what you see in your body functional cells. So this is what people normally sample. But we actually went into the thymus and built a mouse that had two markers, one which was a cell death marker and one was an activation marker. And thanks to this, he was able to sample cells at different levels of this thymic selection step. So first, he's able to sample the cells that haven't been asked any of these positive selection part test questions. So haven't been asked, do you bind to self or not? And then he sees the ones that also have failed. The first test they ask, they're called die by neglect. They die very quickly. And then there's ones that pass. And then there's a differentiation into two cell types. They're called CD4, CD8 if you care. If you don't, just two cell types. And again, then we see the ones that have passed and gotten out of the thymus, meaning they're not supposed to recognize self proteins, and the ones that have failed on getting out of the thymus. So presumably they've recognized self proteins. For the two septides and the ones at the beginning. As I said, either you care in some level about these details, or you just see that we have the failed and the ones that passed and the ones that failed at different stages of this going through this set of things, okay? And since I'm assuming most of you don't actually care, I won't really go into the details of what these markers do, okay? Everybody happy? Basically, the key thing is that normally when you get data, you only see the survivors, right? You see the positive examples. We're also getting the failures, the negative examples, and that's the key of the experiment. Okay. All right, so we're going to try and understand actually selection in the sense that what discriminates the passing and the failing. So, because of what happens, I'll tell. So basically what we do is that we say that the probability of seeing a given sequence at any one of these stages, by which I mean by any one of these subsets that either passed or failed at each stage, is the probability that it was generated in the first place. And here I asked you to, again, trust me that I know what I'm doing, and the probability that it was selected in another stage. So we have a scoring function or an energy function, depending on what your background is and how comfortably you want to call it, right? But basically this tells us the bias that influences the probability of seeing it here, given that they all probably had, come from the same subset. And then we're actually gonna build a linear weight function. We also have models that are machine learning based, deep network based models for this at each stage. We won't use them here just because of scarcity of the data. But if you want, and if you're more familiar with this nomenclature, this is transfer learning, right? We first learn this, which I learned from other data, and then on each of the subsets, I learned a specific selection. Okay. Now, because we're physicists, this of our models actually is as I said, we have a deep learning one, so I shouldn't say that, but we can do an interpretable version of this model where we actually figure out on what is being selected. So since this sequence is built, each receptor sequence is actually built of different segments that I'll call a V gene, a J gene, and the stuff in the middle, which is called CDR3 and is the most diverse. We can actually into this energy function program separately features for this, okay? I'll mention what they look like, so I just want to tell you there's these elements of the model. Yes.
That's on the generative, the PGen, right? Or is it?
That's both.
It's both.
Yeah, so again, I mean, PGen definitely has these components, right? But then we can say selection independently acts, actually, what we say is it independently acts on the length of this whole receptor, independently on the choice of V gene, independently on J gene, and independently on each amino acid in the CDR3, which doesn't mean we assume that the amino acids are independent because PGen couples them. For PGen aficionados like matter. Okay, but anyway, we have a scoring function. All right. So let us run this on the subsets and get some results. I'll go.
Do you really imagine that the energy in the exponential, is it just for convenience's sake looks like binding, or it's really meant to be a?
No, it's more for convenience.
Okay.
Yeah. And as I said, in the machine learning version of the model, it's no longer linear. You have, you allow for correlation. I'll get to correlations between different things because we do test how, let's say, our main reason for going with this is scarcity of data, that I cannot train a full selective model, right? But on the other hand, on data sets where we have more data, we have trained them. And we also know that the difference between the machine learned model and this linear model is pretty small. So I'm getting somewhere more sort of mechanistic with this, but I thought, since I'm sort of introducing myself, I'll try and show different kind of things I do. Okay, so these are different genes. As I said, there's different genes that make these receptors. The blue is the probability of this gene being used in the generative process. And then the different colors are again the different stages. And what I want you to see again is, first of all, that globally, it's how often this gene is used in the generation process that determines how often it's seen in any of the subsets. And second thing, if you take the positive and negative, so this is the one that passed and this is the one that failed, same for the red ones, you see that basically there's no difference, small differences between the positive and negative ones. It doesn't, basically, there doesn't seem to be any difference in the gene. Same thing for length, the probability of generation doesn't change. So all the features we looked at, we don't really see a difference between the passing and failing subsets. However,
for this gene, these genes.
No, these are all the genes.
Oh.
These are all the alpha genes. We also look at it. So there's two chains, right? I'm just showing you an example, but I can show you the same story for everything. So these are all alpha genes in mice. What happens if we put all the features for the alpha chain together, so one of the chains together, and we do a PCA plot, we actually do start seeing some signal, okay? So what we're starting to see is now that they cluster, and they cluster in a way that makes sense. So these are the final points, the ones that passed and failed, the two subsets. This is the starting point, the generation probability. And then basically, so this is the one before the big test, the first big test, and then the split into the two subsets. So there is some signal about the subtypes, but again, the final pass-fail question, that's together. Okay, there is some local clustering, but they're not like in completely opposite paths. But there does seem to be some signal. So let us go on. So what we thought then is, okay, maybe a problem is our model, because we're using the linear model and we really have this non-linear problem, and maybe there's correlations here. So what we did is we tried to find signatures of non-linear selection. And here we used entropy as a measure of diversity. And we basically calculated the entropy of n-grams, which basically means sequences of 3 letters in the sequence. Okay, so we just asked, we forget about the whole biology of the problem, and we ask what is the probability that we make a list of all the sequences of any free letters we see. So like SSA, SSAG, we do a rolling window, ask how often they occur, make a probability distribution, and calculate the entropy of this. Everybody understands what I mean by that? Entropy, everybody's happy, yes?
Why did you pick 3?
We also did 4. We did 1, 2, 3, and 4, and then it becomes again data limitation problems. So no smart reason. Okay. There's a reason why. Okay, so we focus on this very diverse region, which is actually very short in amino acids. It's about 12 amino acids. So again, using 3 to 4 was sort of spanning a good chunk of it. So that's why we felt confident in what we're doing.
Why enforce locality? Why do they have to be contiguous?
Because in principle they don't. But I think we did a non-local version of this, and nothing changed. Okay, but we basically that you have the right idea. There's probably an SI plot with the non-local. Okay. Okay. So basically, if we just look at 1 Ms, so frequency of seeing any given letter, okay, so there's a few points to make here. We have the PGen as the lines, so what we expect from the generation model, and then we have the data, which are just counting the histograms of these NMers, and this is the linear model I just described. And the first thing to notice is that the linear model in any of these graphs and the data always agree. So that's what gives me confidence that the linear model actually is okay. The second thing, just from the fact that there are 20 amino acids, there's a higher bound estimate of 4 free bits. Of course, not all amino acids are used equally, so we already see some selection here. And then we also see selection on the NMers, meaning that there is actually selection on chunks. So there are strong correlations between amino acids, but the linear model again is able to capture that. So nothing really happens. And we do see this decrease in diversity at this final stage, but again, no difference between the passed and the failed. Okay, so, okay, I'll skip this same story. Okay, so then we said to ourselves, okay, in our mind, we're setting up this passed failed question, meaning that I'm thinking about 2 things that should be self-exclusive. So let me see what it would look like on a feature that I actually know is self-exclusive, like length. So if I take a sequence and I have some threshold function for what long and what short means, every sequence, if I run it through this filter, will either be long or either be short, right? Just because, okay? So then I can do that and score them with a long energy and a short energy. Agreed? It's either you're high or you're short. Really, I mean, there's some things in life that are, once you put a threshold, you fall into 2. Okay. So we did this and we scored them, and then we score all sequences with both the short and the long. So a sequence that is really short scores high on the short but low on the long, and vice versa, right? They are mutually exclusive. Doesn't mean when I look at the true positive versus false positives at the AUC curve that it's great, but at least here they truly are anti-correlated. Okay, so now what will it look like on the data? So we do the same thing for all combinations, and I want you to focus on these 2, which is basically scoring the CD4 that failed versus the CD4 that passed, and the CD8 that failed versus the CD4 that passed. Right, so again, failed, passed, they should look like that. They're completely correlated, okay? And just to show you that not everything has to be correlated, the ones that fell off the truck at the beginning, they're actually just a random subset. So that, we're not seeing the signal. Same thing for other things. And when we look at the AUC curve, and this is for these CD4s, we get a very, very bad AUC of 0.58. Same thing for the CD8. These are all the AUCs. So basically, we're not being able to discriminate them. What does this AUC mean, by the way? So what I'm doing is now I have a bucket with the passed, I have a bucket with the failed. I close my eyes and I pick a sequence in random and I ask which bucket did I pick it from, right? That's what the AUC curve means. So I've picked one sequence at random. But now if I pick, what happens if I pick a larger number of sequences, all from the same bucket? Turns out, oops, what happened here? Okay, sorry. It turns out if I pick 30, I already get much better discriminability. And if I pick 100, I have perfect discriminability. Okay, so again, I have two buckets. I pick 100 sequences from the same bucket and I ask, did I pick them from the pass or the fail bucket? And now I can say with utmost certainty which bucket I picked from. But if I pick one, it's random. If I pick 30, I'm already very certain. So there's some wisdom in numbers, right? There's some collective effect. And we don't know what's going on. I don't have a mechanistic explanation of this, but there are cases that we've basically seen things like this. I mean, people have seen things, collective decisions in other immune systems. So Arup Chakraborty again and Marian Kada proposed this kind of effect for self versus non-self discrimination, theoretically. And then Nia Friedman and Michal Polonsky, in a different decision, whether a T cell should become a memory T cell, actually experimentally showed that one T cell is not able to make the correct decision or any decision, but with 30 cells they are. Okay, and so that's sort of what I want to leave you a little bit with a puzzle of how this could be going on mechanistically, where this decision is made. But that's what we're seeing. We're just seeing this feature that if we put 30 sequences together, there is signal, but with this one, there is none.
Could this be, I mean, obviously you've thought of it, so I'm just asking, could it be because in the lab mouse condition, the diversity of antigens that are truly non-self is so small that in some senses, you just have a large ensemble of receptors that are just trying to recognize the same one or two antigens that are really not self? Is it, I guess I'm just asking about the entropy of antigens that are really coming from the environment.
But at this point, when you do this test in the mouse or in yourself, you do it in, again, this pure organ where you only see self. You learn without the negative examples, right? Now we set ourselves up in a setting where we have the negative and positive examples, but your thymus or the mouse thymus doesn't have them.
Yeah.
Yes.
So I don't quite understand what the 30 cells means. So is this that you have 30 cells that all have the same?
No, they don't have the same receptor.
Okay.
They're just from the same subset. There's either from the subset that passed or the subset that failed.
I see. Okay, so it's 30 different receptors, but the receptors are all similar in some sense.
In some sense, but in a way that we cannot figure it out by looking at them.
Right.
Right? So I basically try to show you that there is some evidence, but we can't get at it, right? What we were hoping for is really like a logo. We were saying, okay, you need to have and we didn't think it would just be, this set of letters passes, this set fails, because that would be too easy. Then a virus can easily pretend to be, right? So, but that there is some sort of correlation encoded in this or something like that. And it does seem to be the case, but not in one, but in a bunch. So then we don't know whether actually the decision is distributed and the decision requires many cells, or that this is now speculation. We don't know. So that's what I mean by we don't know the mechanism.
So a possible thing would be that there's 30 cells that all have different proteins.
Yeah.
They all are classified as failing.
Yeah.
And because those are all classified as failing, then we know that those are all kind of bad.
How does it
But
depend on
just a single
NMO?
one isn't enough.
On NMOS.
Yeah, like if you
This
try
is
to do,
back to the energy model, so this is not on NMOS. But that's why I sort of presented the NMOS to get that thought out of your head, that it's not because we have a wrong energy model. Okay, yeah, another thought. So as I said, I'm not gonna give you the answer because I don't know the answer. But actually a former student of ours, Andreas Meyer, who was thinking along similar lines, and he said, well, why isn't it just learnability? Okay, you don't actually have to see every self-protein, but you learn some general properties and then you can generalize. And then he analyzed the data in that sense and showed it's not the case, that there is no learnability in this process. And in fact, learnability, again, from the evolutionary perspective, doesn't make sense, because if Andreas can learn it, then so can a pathogen, right? But yeah. Anyway, as I said, you probably don't care about the system, so I might as well entertain you with puzzles, right? I have like what
we started at 11:15.
So I have like 15 minutes left.
Yeah.
So I can tell you, I have two stories about prediction. Maybe I'll go with the more computer sciencey one.
Sure. What's the other
It's
one?
an older story. It's actually from Andreas Meyer, but it's in a way it's less biological.
What's the other one?
The other one is about memory B cells, and basically when should you diversify and when should you chase after the right answer. Let's. This is maybe more general. Okay, so I'm not going to get to the local objectives. We're going to stay with the local objectives. This is the same idea of shape space, and so the idea is that you basically cover it against various bugs. But of course, bugs evolve, right? And bugs change, even if they don't evolve, because we won't, in this part of the talk, we won't have direct ability, but the bugs that are there and you're trying to protect yourself against change all the time. And the question is, can you build an immune system that can predict the future? So that, you basically, the last year's experience prepare you for the winter that's coming this year in any way. Is your immune system well poised? Because, you don't want to be ready for what happened last year. You want to be ready for what happens now, right? Okay, so how are you going to predict? Well, if you want, I didn't talk about this last time. Okay, not that I expect anybody to remember what I talked about at all, but just for, so if you're going to have any chance of predicting anything, the thing you're predicting has to be correlated. Agreed? Everybody agree on that, right? If you're predicting completely random, non-related numbers, no chance. Okay. So luckily, pathogens are correlated in time because they evolve usually in a continuous way. We can talk about things like COVID and things dropping from out of space. Of course, they evolve too, but just think about something more like this. Okay. Now, the other thing you need for prediction is you actually need to remember. Right? Because if you don't, even if you saw something, but you don't remember you saw it, then, well, then it's just as well as, completely a Markov process or completely uncorrelated random numbers. Okay, so luckily the immune system has a way of remembering. It's called a memory repertoire. And so what happens, so remember that these proliferated because they saw an infection. So that means actually the counts go up, but after the infection, they go back down, but some fraction of them is set aside in what's called a memory repertoire, which is a special subset, which basically has its own nonlinear control of its size. Okay? But just think about it, a cell was useful, it recognized something, gets given a flag saying, hey, you've been to war. Now, why is this biologically useful? Is because the next time something similar comes along, basically either the same bug or something very similar, then this response from the memory repertoire is much faster than this initial response. Yeah.
So is that also mediated by the thymus, or is this some other?
No, so this is all, we're now out of the thymus. This happens on the, on the battleground, which is basically your bloodstream and then lymph nodes. So, yeah, it depends specifically on the cell.
Okay.
There's, will I need to talk about B cells? If I do, I'll get that. But yeah, we're now out of the thymus and in the real world seeing the real bad guys. Okay. Okay, all right. So the question is memory useful? And if it is useful, how should you update it? Meaning if you see this one pathogen one time and then the same pathogen the second time, should you do a proportional memory update? Meaning every time you see it, you keep 2 in memory in my cartoon, or should you somehow modulate it? Meaning the second time you see it, you keep less or more in memory. So we'll try to answer questions like that. Okay, now how are we going to think about it? We're going to assume that there's some.
By the way, Alec,
Yeah.
you don't get the stripes for merely binding. It's some full immune response, and you were the receptor that.
Yeah.
It's more than just you bind. You did bind.
Yeah. It's more than you had to. You really
the
had
whole
to.
proliferation.
So what happens is, I mean, it looks, you have some numbers, of course, that fluctuate, you divide, but when there's an immune response, it really
Right.
does do something like this.
Right.
And then, it goes down. I mean, all of this is,
Right.
of course, messy. The problem is, so if you look at data, there's also some things that naturally have this high level. So looking at just some level will not identify, like I know, there was a response here, if I look at all the, and some of them will just randomly do that. So if I just look at random blips without taking into account the background, I'm not gonna identify a response. So that's a whole other talk. I can tell you how we do that from data, right? But yeah, but it's basically something that has proliferated and gotten all the stripes. Okay, so let's assume we have some ensembles of pathogens that I'm going to call Q of T at a given time. And these pathogens evolve according to some stochastic dynamics where I'll assume I know the evolution operator A. Okay, so I'm just have dt dq A times Q of T. And so they evolve in time. What I'm going to assume here is for simplicity, I'm going to say I have a given fixed subset of pathogens. So imagine my phenotypic shape space filled with, like a lattice. And at every point I have a pathogen, and it's either with concentration 0 or some other concentration, right? I'm not going to make up new pathogens, but they can go extinct and come back. And so I'm just evolving the frequencies. And then those are the bad guys. The good guys is the receptor distribution, which I'm going to call P of T. Of course, the name of the game is for these to recognize this. And I'm going to define an expected cost of an infection, which is the overlap between P and the repertoire and the pathogens, and I'm going to minimize this cost with respect to the receptor distribution to find the optimal receptor distribution for a given pathogen. Okay, so of course the problem is I don't actually know what Q is, right? I don't know what the pathogen distribution is as me, right? I actually know more as a scientist, because I have the WHO and the CDC sampling all of this stuff, but my immune system only sees what I see, right? We'll get to that. So I don't know Q, so I have to estimate it. What does that mean? It means I need to define some believed distribution of Q. and average over that, right? And I mean belief in the probabilistic sense as a meta distribution over the distribution of pathogens, over the distribution of Q, right? So I define this cost and I average over this belief. And so what I'm going to postulate now is what the immune system actually does is it keeps this belief and it stochastically propagates or forecasts this belief in time. Okay, so think about the immune system as basically an ensemble that has a belief about the environment, the pathogens, which it, propagates in time to predict its future state. But of course, once in a while, we do interact with the pathogens. That's the only way we actually have some certainty about what's going on in the world. And that's when we have a collapse in that specific point of the pathogen. We make a measurement and we actually know we can collapse our prior belief into what we've actually seen.
Alex, I'm finding the x-axis of these distributions. I can't even tell a lie about what the x-axis might be. Yes,
Which
it's a frequency. It's a, the P of
The
Q.
y-axis, you mean?
The one above the belief.
Oh,
Just like it's okay. I know it's a frequency, but is it? Yeah.
No, yeah. These are just frequent. Think about it. Don't think about it as an axis. Maybe think about it as a lattice.
Yeah.
Okay. In every point, this will be my Q1.
Okay.
My Q11, Q12, Q21. Okay. And here you think about the frequency, and it can be 0.
Yeah.
Okay. So here you have a number. I mean, a distribution is a bad thing. Also, it's
just a number.
It's a number.
Yeah.
Okay. So you can make it into a square histogram in your head. Okay. Okay. And then I have the dual of this, which is the belief. Right, which is this version of this that the immune system keeps. So again, it has, it has some version of what it thinks Q11 is, and so on.
But it also has to have the representation of the lattice itself.
Yeah.
Which seems like a harder problem.
Let me assume they both live in the same space, which is usually what's assumed. So already when I drew this thing, you didn't complain.
I did. I was complaining in my mind.
Okay.
So
the person to complain to is Alan Perelson.
I see. Okay.
Okay. So, but that has exactly the same assumption, right?
Yeah.
Okay. We can talk about shape space forever, and I would love to, because it's just a phenotypic representation. What is it?
Oh.
No one knows. How to map it on any data?
Yeah.
No one knows. And the first question that you can't get over is, what the hell is the dimension of this?
Yeah.
Right? So I'm drawing things in 2D because nobody can see anything beyond 2D. And actually part 3 of this talk tries to get at what is the dimension of this. Okay. Anyway, so we're propagating our belief, and then once in a while we're seeing pathogens. So we're sampling with some rate lambda, and then and so then basically as this evolves, these do become distributions, right? Because they're evolving in time, and we have they're just spreading, because even if I start with a delta function, it'll spread with time according to my operator. And then if I do measure something, then I'll actually know, is it there or is it not there? So I'll collapse these distributions in these. Okay, that's the general idea. And I'm going to assume that over evolutionary timescales, the immune system has learned the same evolution operator as we have. Okay. And again, we can do this because we have this memory repertoire, so we can remember things. Now, the other remaining thing is that I haven't actually told you what the cost is. So even if at a given time I know what Q is, I still need to define the cost. And here I need to introduce another concept from immunology, which is called cross-reactivity. So Cross-reactivity are basically these things I already drew, these bubbles or spheres or, Gaussian blobs. And what it means is that this receptor is able, the blue one is able to recognize everything in this blue bubble. And this is denoted by this function, which just think about it as a Gaussian for all practical purposes. So if I have a pathogen here, an antigen in the middle, that means it's going to be recognized both by the blue and by the red. So I need to sum over all the bubbles that recognize. Okay, now I'm going to measure time in the number of encounters, and I'm going to say that the cost of the infection increases with the time to recognition. So basically, I have the bug, I'm trying to recognize it, but failing. And as I'm failing, I'm not getting any healthier. There's damage, harm caused to my tissues. So I'm going to say that the recognition events are Poisson distributed. With this probability that comes over the receptors. There's some weighting function, which is an effective cost of infection, which is probably a non-decreasing function of the number of encounters, but it can plateau, and we can explore different versions of it, and we integrate it over all time. And there's a virulence prefactor, because you may care about getting some bugs more than less. Okay, so that gives us the cost for one version of Q, and then we just sum for one antigen, and then we sum over all antigens. Now we have many antigens again. And then we minimize. Okay, so now the results of my minimization. So what I'm going to plot here is I'm going to plot the relative cost compared to not having this memory repertoire, to not having this belief function as a function of the number of pathogen encounters, which is basically the sampling rate times time. And I'm going to do it for different pathogen sparseness. So what I mean by that is basically how often in this matrix I have some input. This is sparse and non-sparse. So first thing you see is that you really get a huge increase. So basically the further it goes away from one, the better. And you have a huge increase for sparse environments. We can also, so memory helps in sparse environments because it's easy to just capture a few very different pathogens. Then we can also plot this for how fast the environment changes. And of course, the more slowly it changes, the similar pathogens you have, and the simpler things are. Okay. But this is what I really want to show you. So we can vary these parameters, but then we realize that actually all of these curves collapse onto one curve. And it collapses as one curve, so this cost, relative cost, if we rescale it by the sampling of non-negligible pathogens, okay, of basically very different pathogens. And then, so we have two, in this collapse, we have two parameters. One is the sparseness, and which is this theta parameter, and the other one is the sampling parameter. So this is reminiscent of basically compressive sensing, that the immune system is able to learn the pathogen environment as long as it's sparse and as long as you're sampling enough.
The correlation statistics are sparse? Like what is sparse?
The pathogens are sparse. That is not yet.
On that lattice.
Yeah. So basically, okay, think about it this way. If I just have a few, if I have a blob here, a blob here, and a blob here, I'm able to zoom in on these, get the idea, because remember what my tool really is. My tool is this cross-reactivity thing. Okay, so I'm going to get to that. I don't really care where the thing is, but with a sparse environment, I'm able to just identify zone of interest 1, zone of interest 2, zone of interest 3. I'm done. The rest are details. Then I can spend the rest of my life learning the details, but doesn't matter. But now if the whole thing is covered, there's like too much, I don't have anywhere to zoom in on. Okay, and that's the intuition. Okay, and so we can compare it to data because one of the predictions of this is that subsequent observations count as less evidence. And this is what we see in vaccination data, that if you vaccinate and you already have a high number of titers, so basically protection against what you're vaccinating with, that means you're here, you don't see a big change after vaccination. But if you have a low number, you see a big change. And another result is that the details of the prior don't really matter, but it's just useful to have some prior. Okay, but I'll say one more thing. And so to sort of finish and answer your question. So what I'm plotting here is what do these optimal distributions actually look like, the optimal repertoires. And now instead of plotting it in 2D, I'll plot it in 2D in a second. I've plotted it in 1D. Okay, the blue lines are where my bad guys sit, my antigen distribution. The green lines is the optimal solution, is the result of the optimization at these optimal repertoires. And the idea is that they're peaked and they're discrete. So although the blue guys are everywhere, because of cross-reactivity, so because of these little Gaussians here, I'm calling them Gaussians, they're obviously not Gaussians, but you know, these little ghost peaks, I do get essentially constant coverage all over space, sort of as I was trying to draw there. And this is the same thing in 2D. I get a speckle pattern. Okay, same idea. Okay, now, quiz time. Two individuals see the environment slightly differently. We're in this room, October, plenty of bugs lying around, okay? Some people are smart, wear masks, okay? The rest of us, we're essentially sampling the same environment, but we're sampling it slightly differently, right? Just through random. So say you're the blue one, I'm the yellow one. That's how I'm seeing the environment. That's how you're seeing the environment. Should we have the same optimal immune distribution against this environment or different ones? Very good. Okay, I see votes for different, right? Everybody?
What do you mean by, you could have statistically the same, like an expected value that's the same?
Yeah, that would be the same for me. I mean in terms of overlap. If I calculate the overlap between mine and yours, should we have the same? Okay, that's basically, maybe I'll answer the question.
Okay.
Basically that, okay. So we can have very different repertoires, meaning that we actually, we all get the peaks, but we get the peaks in different positions. And that's sort of shown here. Okay, but so the similarity, so if you want to call that statistical or not, between our repertoires goes down to zero very, very quickly as a function of how differently we see their environments. Although what we're seeing is still very similar and we both have perfect coverage against both of them. Why? Well, because of what I was explaining here and here. So these receptors now are actually placed in different places on the lattice, which is what the grid lines are telling you. But from afar, they still cover space. Okay, and so we basically can have very different solutions. And then if we go back to data, we can calculate the overlap at the sequence level between our repertoires. And we can actually predict it perfectly, which is another talk. But if we look at the numbers, of course, it depends on sampling, but the numbers are very small. So and we're sampling here of the order of millions of receptors from one person, and the sharing of the order of 100. Okay, and that's because the ideal match doesn't really matter. Okay, and I'll stop here.
Questions?
I didn't. At least people passed the quiz, so
So small children have vastly different immune systems than adults, right?
I'm being recorded.
I'm being overly simplistic, so I'll just say.
Well, yes and no.
See what Alex thinks matters.
Yes and no. So small children, we don't sample for ethical reasons. We sample small mice.
Right.
And so what happens is, until you're born, the diversity of your repertoire is very reduced.
Right.
Because the way it works is that the part I didn't explain is that this part in the middle, which I called CDR3, is actually based on random non-templated insertions, and the enzyme that does these insertions is down-regulated until you're physically born.
Okay.
And so then it starts being upregulated, and you start having more insertions, which generates the diversity. But you should also think about it as basically you're starting to generate. So in utero, embryos do generate some cells, but by the time you're born, you're nowhere near having actually generated a full repertoire. So then you sort of go full speed and fill up this empty bag without having seen anything really. And on top of that, if you get antibodies from your mother, so important, you get antibodies which are soluble receptors and not actual cells from your mother. That means that when they'll wane and they have a short time lifetime, that immunity will go away. But you're basically in the beginning, you're probably protected by these, and then only do you have to start doing your own thing. But you're mostly naive because you haven't seen anything. So one thing that changes through life is that the proportion of your naive, so they all have their own size control, but the relative proportion of the naive to memory changes. And as you get older, you get smarter and have more memory, but you also, your thymic output, so the production of new cells goes down. And older means basically end of puberty. Right, so then, the sort of trends continue, but until puberty, yes, there's all this dynamics going on that, you're still training your memory repertoire, you're basically starting to push out these non-diverse cells, and in that sense, they're different. But also that's why they go around licking each other, because it's good.
Good. I'm actually suffering it right now. Okay, any more questions? Yeah, Danny.
Maybe this question doesn't make total sense in the context of what you just presented, but I guess I'm curious, is there a cost to having more memory of past infections? Like, I didn't see it in your cost function. Your cost function is the cost of the wrong expectation. But where is there embedded in there a limitation in like the amount you can remember?
Yeah, that's part 3 of the code. So this is another cost function where we have this is basically answering more the question of, well, let's go to the one bug that we're actually trying to track that is actually physically evolving according to sort of more realistic update mechanisms, right? So I want to, and the cost function is basically this. First of all, you have some background, some baseline from naive innate immunity, and then you have existing memory. And actually, when you get infected, it's not, this whole process starts going, but these are new memory cells that, well, okay, maybe this is wrong picture. But initially what happens is your existing memory cells, even if they're imperfect, they're the ones that are trying to fight the infection. As a result of an infection, you'll produce new memory cells, but they'll be useful for future infection. So this is what we try to explore that with precisely. And
so but storing those for a future infection, does that come at the cost that you have then fewer naive cells? Is that
no, it comes at this. So again, think about this as 2 bags. So you have the naive bag and the memory bag, and you do size control, homeostasis in each one of them separately. Then what I was just saying here was just some overall trend of how these sizes go. But that means that basically to remember new things, you have to forget something, but in the memory one.
that's
and actually, one of the answers to I didn't talk about that, but one of the things we also learn here is that if you're gonna remember something, you need to forget. So forgetting is an inherent part of learning.
Let's thank Alex again.